basic structure of the following molecules. A saturated fatty acid, 16 underscore or 16 0, and then N-hexadecanoic acid. Okay, so this number right here tells you, let's see, this number right here, okay, I guess not, 1 second. This number right here tells you how many carbons, and this number tells you the number of double bonds. So for a saturated fatty acid, I'm going to draw it the way that it's in your key. But basically you start off with a, let's see, a hydroxyl group, carbonyl, and so this is your first carbon, and then, so this is your second, this would be your third, so 1, 2, 3, and you have to draw 16. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, and 16. Okay. Okay, so I just wanted to point out really quick that this in A is a saturated fatty acid because there are no double bonds. Okay, for B we have an unsaturated fatty acid. Okay, the fact that they're saying unsaturated, that means that we're going to have double bonds. So 16 carbons, 1 double bond. Where is that double bond going to be? It's going to be on carbon number 9. That's what the triangle tells us. And then how is the double bond going to be? Is it going to be cis or trans? And it tells us right here, cis. So this is what cis looks like, and then this is what trans looks like. Okay? So we're going to go ahead and start the same way. We have our hydroxyl, carbonyl. This is carbon number 1. So this is 2, 3, 4, 5, 6, 7, 8, 9, 9. Okay? So let's do that. 2, 3, 4, 5, 6, 7, 8, 9. Okay, so now we have to draw a cis double bond. There we go. And then we just keep going. So this carbon 10, 11, 12, 13, 14, 15, 16. Okay, and it's that easy. Okay, so number 1, draw the basic structure of the following molecules. C. The structure of glycerol and the resulting general structure of triacylglycerol with 16 carbons, 0 double bonds at carbon 1, 18 carbons, 1 double bond at carbon 9, and that's carbon 2, and 18 carbons, 3 double bonds at these locations at carbon 3, and circle the ester bonds. Okay, so first of all, you need to be really familiar with glycerol. Okay, so you should know what that looks like. There you have it, that's glycerol. So what is a triacylglycerol? That's what you're supposed to be drawing. So that's super easy. That just means that you have a glycerol molecule with 3 fatty acid tails attached. And then all of this information right here, like carbon 1, whatever's going on, and then whatever's going on at carbon 2, and whatever's going on at carbon 3, that's actually talking about glycerol's carbons. So this would be, let's go ahead and do, this would be like, okay, this would be carbon 1, 2, and 3. So they're just telling you, they're describing each of the tails. So let's go ahead and draw that. So you always draw your oxygen, and then when it binds to the fatty acid, it'll look like this. And so the first one said it was 16 carbons with 0 double bonds. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So there are 16 carbons with no double bonds, okay? The next one, let's go ahead and draw our structure. Okay, and the next one says 18 carbons, 1 double bond at location 9. So here's 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so at location 9, and okay, so it doesn't say cis or trans, so we're just going to draw it like that, because that's what it is on the key. And then, so you have to get all the way to 18. So that's 10, 11, 12, 13, 14, 15, 16, 17, 18. So that's 18. And then the last one is, let's see, at carbon 3, we have 18 with 3 double bonds at locations 9, 12, and 15. Okay. Okay, so that's 1, 2, 3, 4, 5, 6, 7, 8, 9. So that's where the first double bond occurs at 9. So there we go. And then 10, 11, 12. And that's where the next one occurs. Okay, so that's 12, 13, 14, 15. And that's when the next one occurs. 15. But it has a total of 18 carbons. So that was 15, 16, 17, 18. Yeah. Okay. So we can actually number those. I know some students struggle with the numbering, so we could just start here. 1, well, let me do purple. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, and 18. Okay, and that's it. Okay, and it also tells you to circle the ester bonds. So what does an ester bond look like? Here we go. Okay, so this is the ester bond. So remember, this is a carbon right here. The carbon has a carbonyl and then the oxygen. So there you are. Okay, D, a glycerophospholipid. Draw a box around the polar head group. Okay, let's draw a glycerophospholipid first. So let's see. Okay, so notice that the last tail doesn't have a fatty acid tail, it has a phosphate instead. That's why it's a glycerophospholipid. Okay, and so you can draw just like any phospholipid. So there's some general trends here, like the first carbon usually has a saturated fatty acid tail, which that's what we have here because there's no double bonds. And the fatty acid attached to carbon 2 is usually unsaturated, so you will usually see like one double bond or more. So we're just going to go ahead and draw one. So that's generally what you will see. And then attached to this phosphate group is where you will normally have the polar head group substituent attached. Draw the basic structure of the following molecules. 1E, sphingosine, and a general structure of a sphingolipid with phosphocholine as the polar head group. Draw a box around the amide linkage. Okay, so we're going to go ahead and start with this right here. Let's see. Okay, we have our 3 carbons, then we have our hydroxyl group. Okay, then we have and then the second carbon has the amide linkage. So that's the nitrogen, carbonyl, and the hydrogen. And then the rest of our carbohydrogen chain. So this is the amide linkage. And then on carbon 3, we have our phosphate group. And then 2 CH2s, because they want phosphocholine. So that's phosphate CH2, CH2, and then N, positive charge, and then there's 3 methyl groups attached. And that's it. Okay, so here we have cholesterol, and they want us to draw the polar head group. So that's right here. Remember, alcohol groups are polar because the electronegativity for oxygen is a lot greater than that of hydrogen. And this group over here is nonpolar. So if you're being asked like which one of these will face the cytoplasm, remember that the cytoplasm is mainly water, and water's polar. So this guy will be near the cytoplasm or facing the cytoplasm. Question number 2, what is the most significant chemical difference between triacylglycerols and glycerol phospholipids that leads to their different functions? Okay, so for this, it's kind of just like definitions. Triacylglycerols are nonpolar. They're hydrophobic molecules that can be stored in specialized non-aqueous cellular compartments. And glycerophospholipids are amphipathic. They can serve as structural components of membranes, which have hydrophilic and hydrophobic regions. 3. Glycerophospholipids, sphingolipids, and sterols spontaneously form microscopic aggregates when mixed with water. A. Explain the role of hydrophobic effect in the formation of those lipids or those lipid aggregates. So again, it's just like a definition, things that you just have to know. Aggregation causes the hydrophobic moieties to cluster together. Remember, they're trying to get away from the water, so they cluster together with each other, while the hydrophilic groups interact with the surrounding water. This minimizes the number of molecules of water that is needed to form an ordered shell around any exposed hydrophobic moiety of water interface. This results in an increase in entropy and therefore generates the lowest energy environment. (3b) Draw a lipid micelle, a bilayer, and a vesicle, and explain the differences between them. So here we're talking about amphipathic lipid aggregates that form in water. So there's different kinds. There's 3 different kinds. And so the first one is the micelle. And so that looks like this. It only has 1 tail. So individual units are wedge-shaped. The cross-section of the head is greater than that of the side chain. So what they're talking about here is this. The cross-section of the head is bigger than that of the side chain. So it looks like a wedge. Okay, that's the shape. And when a lot of them aggregate together, the way that they look is something like this. So this will be the center. And then you'll have a lot of the little tails coming out. And then you'll have the head groups. Okay, and that's a micelle. And what's important to note about this is that at the center, there's no water. It's literally all the hydrophobic tails. All the hydrophobic tails at the center. And the polar head groups are on the outside. They're facing the water. Okay, so now we're going to go ahead and draw a, a bilayer. So the individual groups look like this. There's the polar head group, but these guys have 2 tails. Okay, so the shape is more of a cylinder, like that. And then whenever these guys form, they form like this. Okay, and then the same thing at the bottom. And this is how lipid bilayers are formed. Okay, so that's a bilayer. Lastly, we're going to draw a vesicle. And when a 2-dimensional bilayer folds on itself, it forms a closed bilayer. And this is a 3-dimensional hollow vesicle, a liposome. And it encloses an aqueous cavity. So what that looks like is a circle of polar head groups. Okay. Where there are 2 tails. And then over here, you have polar head groups also. And then their little tails pointing this way. But because it folded in on itself, what ends up happening is that you end up getting water, like an aqueous cavity of water right here. So there's water on the outside, and then there's water on the inside. And this is a vesicle. Why can't triacylglycerols be significant components of the lipid bilayer? So if we remember what a triacylglycerol is, it's just this right here. So you have your glycerol with your carbon, so that's carbon 1, carbon 2, carbon 3, and then 3 fatty acid tails attached. So one thing that we don't see here is any polar head. So there's no polar head groups. And you need that for the bilayer, right? Because a portion of the bilayer is going to be facing the water in the cytoplasm or cytosol. So if you don't have any of that here, then this can't be good for the bilayer. It's got to perform a different function. Does the phosphatidylglycerol head group of cardiolipin project out of the lipid bilayer like other glycerophospholipid head groups? The answer is no, because the head group has 2 acyl chains that are buried in the interior of the bilayer. So cardiolipin is one of those molecules that you should practice drawing a few times. Definitely get familiar with it, learn to identify it. Okay, which of the following is not correct concerning the structure given? All right, so the first thing I like to do is start labeling stuff. So this right here are CH2, CH, and CH2. That's glycerol, okay? Okay, here we've got phosphate. Okay, then over here we have choline. Then this region right here, we've got CH2, we got 7 of those, and then CH3 right here. So this whole section is nonpolar. Okay, so let's look at our answer choices. Which of the following is not correct? So it is a component of biological membranes. It is amphipathic. It is a sphingolipid. It is a glycerophospholipid. It is phosphatidylcholine. Okay, so let's see. Phosphate, and that's nonpolar. I mean, that's polar. And then over here on the left, we have nonpolar. So the fact that we have polar and nonpolar, that means we have amphipathic. Okay. We have glycerol right there, and we have phosphate. Phosphatidylcholine. Okay, so the answer, the only one that doesn't fit, is a sphingolipid. So if you practiced drawing your sphingolipids earlier, then you would know that that one doesn't fit. Which of the following membranes would be the most fluid? So I gathered some rules for you down here in red. If you have low temperature, you have lower fluidity. Cholesterol helps to increase fluidity in this case. If you have higher temperature, you have higher fluidity. Cholesterol helps to reduce fluidity in this case. If you have a saturated fatty acid, the fluidity decreases. Okay, I had to pause and I don't remember where I was. So I'll start with unsaturated. If you have an unsaturated fatty acid, that means you have double bonds and your fluidity increases. If you have longer tails, you have less fluidity, and shorter tails is increased fluidity. So let's go ahead and jot down which ones are for increased fluidity, because that is what the most fluid. Okay, so that's what you want. So let's see, low fluidity, higher. So higher temperature is higher fluidity. Unsaturated, so double bonds, that's, no, saturated. No, unsaturated, fluidity increases, okay? And then we want shorter tails for increased fluidity. Okay, so A bilayer made of lipids with unsaturated. So unsaturated, that's good, that increases fluidity. Let's see, there's 18. And B, a bilayer made of lipids with saturated 18 carbon. So saturated is less fluid. A bilayer made of lipids with saturated 16 carbon. Okay, and then a bilayer made of lipids with polyunsaturated. So that would be more fluid, and then 16 carbon. That's a shorter tail, so it's got two factors going for it. It's a shorter tail, which is more fluid, and it's unsaturated. So that's more fluid also. And then E, all of the above are equivalent in fluidity. No. So our answer is D. Which of the following statements is consistent with the structure of biological membranes? All membrane proteins are integral and associate with the hydrophobic region of the membrane. So no, not all membranes are integral, and not all of them, or not all membrane proteins are integral. So we have to be careful when we see all, and then there's an assumption about all proteins. So that will usually be false. B, both proteins and lipids readily undergo transverse diffusion from the inside to the outside of the membrane. No. C, membranes are symmetric. Okay, no. D, the membrane lipids self-assemble to form the lipid bilayer. So yes, that does happen. And E, a biological membrane consists of proteins sandwiched between two layers of lipids, which are referred to as a lipid bilayer. No, that's not the definition of a lipid bilayer. Okay. Number 7, explain why the extraction of integral membrane proteins requires the use of detergent, whereas peripheral membrane proteins can be released from the membrane by treatment with high salt or changes in pH. So make sure you have your definitions memorized. You should know what an integral membrane protein is, detergent, and a peripheral membrane protein. If you don't have those definitions jotted down, then look them up. And then this is your explanation. Integral proteins interact with both the hydrophobic heads and the hydrophilic tails of the membrane, meaning that all four types of non-covalent interactions are occurring. Therefore, a detergent must be used to break up the hydrophobic interactions. Peripheral proteins are not associated with the hydrophobic region of the membrane. Only the polar heads of the lipids or aqueous domains of the integral membrane proteins through ionic interactions. Those ionic interactions are disrupted by high salt and changes in pH. So when there's more salt ions available to compete for those interactions. And when you have higher pH, that's the charge of the acid-base groups that is changing.